Good afternoon, everyone. Welcome to the Wednesday afternoon lecture series. I'm Alexandra Nita-Lazar, Chief of the Functional Cellular Network section in the NIAID Laboratory of Immune System Biology, and I'm thrilled to be introduced today's WALS speaker, Dr. Benjamin Garcia, who is conducting what I think is some of the most exciting work today on high-resolution mass spectrometry to explore cellular signaling, epigenetic mechanisms, and chromatin regulation. But first, I have a few announcements. The first concerns continuing medical education credits. The CME code for today is 50129. I'll repeat that, 50129. Also, for those of you watching remotely, you can submit questions at any time by clicking on the button just below your videocast window that says Send Live Feedback, and click on that, type your name and question, and we'll relay this to Dr. Garcia at the end of the lecture. For those who are present here, there are 2 mics in the aisles that you will be able to use at the end of the lecture. Please note also that today's lecture is the final in this year's WALS season. We will take a 2-month break and begin a new season on September 11 with a lecture by Dr. Eve Marder of Brandeis University on neuronal networks. Okay, our speaker today is Dr. Benjamin Garcia. He's a Raymond H. Witkoff Distinguished Professor of Biological Chemistry, Professor of Biochemistry and Molecular Biophysics, and Head of the Department of Biochemistry and Molecular Biophysics at Washington University School of Medicine. Dr. Garcia received his PhD in analytical chemistry from the University of Virginia, Charlottesville. He then spent his postdoctoral years at the University of Illinois, Urbana-Champaign, under the mentorship of Neil Kelleher. From 2008 to 2012, he worked at Princeton University, where he was assistant professor, and then he joined the University of Pennsylvania Perelman School of Medicine as director of Quantitative Proteomics, and in 2018 became the John McCrae Dixon MD Presidential Professor. And in 2021, Ben moved to the Washington University School of Medicine in St. Louis, where he attained his current title as Raymond H. Witkoff Distinguished Professor. Ben is very active in the proteomics community. For example, he is part of the nominating committee for the Human Proteome Organization, HUPO, chair of the Diversity, Equity, Inclusion, and Respect Committee for the American Chemical Society, and a mentor for the ASBMB's NIH-funded program called Maximizing Opportunities for Scientific and Academic Independent Careers, or MOSAIC. He's also associate editor of Analytical Chemistry and an editorial board member of numerous journals in our field. He has received several honors along the way as well. As you might imagine, he was named a highly cited researcher by Clarivate in 2022. He has received the Human Proteome Organization Discovery in Proteomics Science Award and was elected as Fellow of the Royal Society of Chemistry. Benjamin's primary research focus has been to characterize the role of post-translational modifications, or PTMs, on proteins involved in human biology and disease. He's especially interested in understanding how protein and nucleic acid modifications mediate their canonical functions and regulate nuclear processes. That said, his research group has broad expertise in many areas, including chemistry, particularly in the area of mass spec-based analytical chemistry, informatics, biochemistry, genomics, cell biology, and molecular biology. As he likes to joke, his underlying goal is to bring mass spectrometry to the masses, and he and his research group has done just that with the development of tools and techniques that have allowed for high-throughput quantitative mass spectrometry analysis of histone PTMs and chromatin structures. He also exemplifies that by his relentless advocacy during the yearly JBC Method Madness, where several very, very good methods compete to become a method of the year, and all years, I think, since it's been conceived, at least several years in a row, mass spectrometry has won, and it won against methods such as CRISPR, flow cytometry, etc. So it's not a joke; it's a serious competition. Yeah. In more recent years, he has been upping his game with translational research and deeper connections to human disease. So the title of Ben's talk today is Quantitative Proteomics for Understanding Epigenetic Mechanisms. Please help me welcome Dr. Benjamin Garcia.
Okay, great. Thank you, Alexandra, for just a great, wonderful introduction and being a fantastic host. It's been a wonderful day of meeting many incredible scientists and trainees here. I've really enjoyed it so far. And so what I'm hoping to do today is give everyone a little bit of kind of a taste of some of the technology and approaches that we've been developing in the last couple of years and how we're applying them. One to a problem in cancer epigenetics and probably a little bit more of things we've published and also a newer story that's emerging as well. And I guess I'll probably use, well, let me see. If I use this, will people online be able to see that or no? It should be. Really? Oh, okay. Okay, great. So, yeah, right here, you can see it. Okay, you can't see. All right, then I'll use this. And so, yeah, maybe first, before we get into the technology and the applications, just wanted to give a couple slides on the background. And I know not everybody works on chromatin and epigenetic mechanisms. That's okay. Nobody's perfect. But just to give a little bit of background, we have meter amounts of DNA that have to be compacted into a very small area in the nucleus. And the way that nature does this is by taking the DNA and winding it on a ball of proteins called histones, very similar to winding a string on a spool. And so you have two copies of each of the core histones, H2A, H2B, H3, and H4, and about 146 base pair of DNA make up the nucleosome, the building unit of chromatin. And the nucleosomes are further put together and essentially folded into chromatin fiber in our chromosomes. And it was thought that these histone proteins were just very structural components of chromatin, but over the last 2+ decades, it's been found that these histones that have a huge array of post-translational modifications are dynamic proteins that help regulate gene expression patterns. And so kind of fast forward now by many methods, including mass spectrometry, several post-translational modifications on these histones have been described. You can see here a whole host of many different chemical modifications across many sites, essentially the entire protein of these histones. However, definitely I would say, the majority of the modifications are found on the N-terminal regions of the proteins, the more high stoichiometric modifications. But you can see here, there's some sites that have multiple modifications, 10, 11 different types of modifications on the same residue. So it's a very complex language and one that keeps an analytical chemist such as myself really busy with detection of novel modifications and identifying quantitatively when these modifications change from one state to the other, and also identifying which combinations of modifications occur together as well. And it's these histone proteins that, along with DNA methylation and non-coding RNAs, make up these epigenetic mechanisms. Epi is Latin for on top of, so these are all the mechanisms on top of genetics that regulate gene expression. And then these histone post-translational modifications, again, kind of shown just in cartoon form here, some of the more abundant modifications, such as methylation and acetylation. But these modifications have, by biologists, more or less one at a time, been linked to many different important processes, especially turning genes on or off, and also in DNA damage events and things like that. Histone acetylation, in general, is linked to gene activation. Histone methylation, it's a little bit more complex. There's different degrees of methylation, and it can also be linked to both gene activation or gene silencing, depends where the modification is found. And so, for example, this methylation right here on lysine 4 is found on active genes in the genome, promoters of active genes in the genome. This methylation on the same histone protein, just a few amino acids down from this other modification. Let me see. Oh, I lost the pointer. No. Okay, can you? Okay, I'm not sure if I have a pointer anymore. Oh, there it is. Okay, okay. And so this modification, same methylation on the same protein, just a few amino acids down. This has actually been found on the heterochromatic regions of the genome. And it's because of this relationship that a lot of theories and hypotheses have kind of been put forward to try to explain how these modifications may regulate gene expression. And one of these is the histone code or PTM code kind of hypothesis, which states that these modifications well, if you think about it, if there is a code, there's got to be a way to interpret this code. And it's thought that these modifications act as a code by bringing proteins that then read or interpret the code to specific parts of the genome. And so it's thought that these modifications act as a platform for recruiting these proteins to activate genes. And in support, several proteins with specialized domains have been found to bind specific spots on these histones and in the genome. And there's a lot of interest right now in kind of understanding, especially in human diseases, how these epigenetic mechanisms can go awry. Now with, a decade's worth or more of large-scale genomic data sets, several mutations that affect epigenetic machinery that modify histones or proteins that interact with histones or even the histones themselves have been found to be mutated in various cancers. And because of this, several companies, including very large companies, have epigenetic oncology divisions. And there are smaller companies that only focus on this, with epigenetic inhibitors kind of making their way through clinical trials and FDA approvals. And so there's a lot of interest in understanding if, what you're seeing in terms of transcriptional changes involves epigenetic mechanisms in human disease and biology. But I always kind of tell people that you really know you're working in a great field when it makes it into blockbuster movies. And so here's a film still of one of the Spider-Man movies from a long time ago. And so you'll see all the science on the board of everything you need to know to be able to get superhuman power. You might recognize some phrases or schematics and things like that. And if you look right at the bottom here, there's something that's really familiar. It's a nucleosome with histone H3 K27 methylation on it. And so whoever knew that they had to put the most important science on this board for this movie knew they had to include epigenetic mechanisms. And so there's a lot of different ways to look at histone modifications. The most common approach is using site-specific antibodies, which if you've used antibodies, can be both a blessing and a curse. If you have a good antibody, you get the one signal. This modification actually doesn't exist in yeast, so this is what you would see, and you shouldn't see it in the recombinant protein. But see here, you'll see, kind of cross-reactivity with another protein that has a similar epitope. So you'll see a couple other bands here, and this antibody right here is recognizing recombinant histone H3, so it's not even recognizing a modification. And so this is why biologists only show the little sliver of the Western blot, not the whole thing, because then you'll see all the other proteins that it's not reacting with. So you have to be really careful, especially with such modification-dense proteins such as histones, to really understand what epitope the antibody is recognizing and to not get fooled that it's maybe recognizing another histone or something else. The other issue as well with antibodies and histone modifications is that I showed you how many different modifications there are on these histones. And so you can imagine that some of these modifications might actually block the antibody from recognizing the intended site. And so here's an example of this. It's just a peptide ELISA with this antibody against serine 10 phosphorylation, and these peptides that all have serine 10 phosphorylation, these two have methylation acetylation next to it. This antibody is really good at recognizing the phosphorylation by itself, but struggles to recognize the duly modified peptide with either the methyl or acetyl group. And this does exist in vivo quite abundantly. We can detect multiple modifications by mass spectrometry much more easier than you can with antibodies. And we could do this quantitatively as well. And so now, over my lab's existence, we've been developing a lot of methods using quantitative mass spectrometry, quantitative proteomics, to essentially understand a lot of different facets of histone PTM biology and protein PTM modifications as well. So just trying to create very quantitative, high-throughput methods to analyze as many histone modifications at a time. trying to understand which combinations of modifications occur together, because our data and others show that one modification on a histone at a time just doesn't exist. It's always in combination with something else. But if you're only using antibodies, you're only recognizing that one modification. We've developed a lot of metabolic labeling approaches, combining that with proteomics to be able to understand the histone dynamics and get at the kinetics of these modifications, because these modifications are not static. They're constantly being put on and off, and also combining genomics with the downstream proteomics readout so that, hopefully one day we can get to the point where we're pulling out our favorite gene and identifying all the proteins and histones, modifications that are on that gene, in both kind of the physiological and disease state. So for this first part of the talk, I was just going to kind of give an update on some new approaches and new technology that the lab has been kind of evaluating over the last year plus. We're always kind of on the lookout for new technology that gives us some advantages and allows us to do things that we weren't able to do before. And one of the crutches of quantitative proteomics, except for in the last year or so, was that it's not very high throughput. It takes a long time running 1 hour or 2 hour gradients on these instruments. And our gold standard kind of nano-LC run on an instrument to detect histone modifications would take 60 to 90 minutes. So it's definitely not a high throughput approach. But now you have a lot of instruments that are coming out. They're based on different mass analyzers than Orbitraps, which are high resolution instruments, that are much faster scanning instruments. And so we were kind of interested to evaluate a few of these, and one we actually ended up really liking, not just for the high throughput abilities, which I'll show you in a second, but also because this instrument is equipped with a different fragmentation mode, electron activated dissociation, which allows us to do some things that we can't do in terms of fragmentation for these histone proteins. And I'll show you how we're kind of using that in different ways as well. And so right about the time we got this instrument, our friends at SciEx, which, I'll disclose right now, I don't have a research agreement or anything like that in place. We're just, happy customers. But our friends at SciEx published this paper saying that essentially could quantify about 1,000 proteins per minute on this instrument. It's kind of hard to read this, but this is a 5-minute gradient, if you can kind of see that. So you're talking really, really fast quantitative proteomics, almost kind of too good to be true. And, honestly, when stuff like this comes out, I usually kind of say, oh, well, I don't know if I want to believe this propaganda. Like, I need to do it kind of for myself. So kind of got all the parameters from people at SciEx and was able to kind of reproduce this. I had a postdoc in my group, now my lab instrument manager, who was able to reproduce this. So this is our data here, again, 5-minute gradient. And essentially we get anywhere from 4,500 to 5,000 proteins in 5 minutes, detected with 3 to 4 different peaks across the points across the peak. And so this is actually a really, really fast instrument. And now this is kind of our QC. Before we would run either a whole proteome and kind of run a 2-hour gradient or maybe a few synthetic standards and run like half hour. Now we run a 5-minute QC before every batch of samples. If we don't get 4,500 to 5,000 proteins in 5 minutes, the instrument may need to be cleaned or something needs to be adjusted. So we were quite happy with that. And as I mentioned, our gradient for normal nano-LC-MS analysis of histone modifications is about a 55-minute gradient, maybe close to like an 80, 90-minute total run. And so we created some really quick gradients to see if we can get very similar data from just these much faster high-throughput runs. And so the big difference here is that we're using microflow LC. So for those that are in the proteomics field, we're using 5 to 10 microliters per minute flow rates instead of 100 to 200 nanoliters per minute flow rates. We wanted to evaluate these. And so we set up a whole data independent acquisition. Let me get through some of this transitions mode where we can kind of optimize the windows that we're assessing in the mass spectrometer a little bit more dynamically. There's some areas that don't have a lot of modified histones, so you could use bigger windows. And instead of kind of just going through with equal windows, which we did before on the Orbitrap instruments, because that's about as fast as we can go through, here we can kind of take the time to tailor and optimize the windows. We know the bins where the most modified proteins from these histones come out at. And when we looked at the quantitative data, we were quite happy with this. And so you have the 55-minute normal nano-LC run, and then you have these crazy fast 5-10-minute gradients or runs. And you can see here, peak shape looks really, really strong. When you have a peptide that has multiple lysines and you have a modification such as an acetylation, the modification can come out on these different lysine residues, and you often have these peptides that overlap retention time-wise. But you can see here, you can easily look at the transitions and tell, where the modifications are found, even if they partially overlap. And so we get enough points across the peak to really be able to differentiate these isomeric peptides. We can do this really well, and we've kind of developed a few softwares to be able to handle this data as well. In terms of total number of peptides identified or the CVs of these peptides, they're pretty consistent across all these different types of runs, kind of our gold standard nano-LC run, hour plus. And then the 10 and 5 minute. 5 minute might be a little bit quick, so we kind of like the 10-minute kind of gradient. But you can see here, very, very solid across all the way in terms of the numbers. Incredibly robust instrument as well. So here you have 100 repeat injections of the same sample just right on top of each other. And you can see here retention time and number of peptides is pretty consistent across the large majority of these runs. And so now we can get to the point where we're able to work with our clinical colleagues. I would have many clinical colleagues at Penn and now at WashU that would come up to me and say, hey, we've been following these patients for the last 5 years, taking samples, blood samples every month, and we have thousands and thousands of samples. We would love to profile for histone modifications. I'd be like, I can't run that many samples. Like, it would just take too much instrument time. But with now, this 10-minute gradient, maybe about 17-minute total run, we can get to the point where we're running hundreds or thousands of samples in a week, and we can take on some of these very large-scale clinical projects or these or population type of studies as well. I mentioned this electron-activated dissociation. This is very, very different, something that not all instruments have, the ability to use electrons to fragment peptides. Most instruments use collisions with an inert gas. And so here you can see if you increase the energy, you get the fragmentation. It gives you very, very complete coverage in between almost every amino acid, so you can tell exactly where the modification is. But we found that we actually were able to use this in a different route to help solve a really difficult problem. And so I had a collaborator at Yale, Matt Simon, who said that by other methods, he had evidence that a lysine residue could have an acetyl group and a monomethyl group together at the same time. And so I've never heard of that, having two modifications on the same lysine residue at the same time. And he wanted to know if our mass spec methods could help him get some more direct evidence of this. The one issue here is that the mass of an acetyl methyl is the same as the mass of another modification, this fatty acid modification called propionyl, same elemental composition, so you can't really tell them apart just by mass. When we fragmented these peptides together from his samples, we saw that there was two slightly overlapping peaks, but when we fragmented them together, basically gave us complete coverage. And so since these are the exact same isobaric mass, we couldn't tell the difference. But the nice thing about this electron-activated dissociation is that it's very tunable, and you could crank up the energy and do different things that you can't do on other instruments that have electron-based fragmentation modes. And so we cranked up the energy to the point that we were breaking off the side chains of the amino acids. And when we broke off the side chains of this lysine that had the acetyl methyl or this side chain that had the propyl, then by the fragments of the side chain, we can tell that this actually was an acetyl methyl when we compared it to synthetic peptides. And this was the propionyl group. And so this was the first evidence of actually having an acetyl methyl modification together on any protein at the same residue at the same time. And so now I kind of just want to get into two projects to show you how we're using this technology. This one, we've gone a little bit farther and published. We've been interested in soft tissue sarcomas for a long time. There's a whole host of mutations to many different types of soft tissue sarcomas that either affect chromatin remodeling proteins, such as SWI/SNF or polycomb, or have these really kind of interesting fusion proteins that are created where you have a histone-modifying enzyme that's fused to a zinc finger protein, transcription factor or something like that. and it's not really clear, the molecular mechanisms for how this leads to the progression of the cancer. And there's even histones that get mutated themselves. So you can imagine if you had a lysine residue and now you have a mutation to something else, you just wiped out methylation or acetylation across the genome on this histone. So there's all these really interesting types of mutations that occur. And one of them that we've been very interested in is this malignant peripheral nerve sheath tumor, MPNST. So as its name implies, it's a sarcoma of peripheral nerve origin. It could happen both sporadically and syndromically, and so most patients have an underlying benign mutation to the NF1 gene, neurofibroma gene, and then we'll get the secondary mutation that will lead to cancer, but older patients can sporadically just also get those secondary mutations and progress to cancer. It's a pretty rare tumor, maybe several hundred a year that are described, but it's very aggressive, so less than 50% 5-year survival rate. There's not a lot of options to treat, basically just surgery and radiation, and about half of the patients have a relapse. There's a lot known about the mutations that lead to MPNST, but about 80% of patients have a mutation to one or two components of the polycomb-repressive 2 complex. And so this is an enzymatic complex that methylates histone H3. It creates a trimethylation at histone H3. And in fact, this is how pathologists differentiate different MPNSTs by staining for the K27 trimethyl. So here's an MPNST with polycomb-retain. So it has a different mutation. You can see the brown staining of the histone H3 methylation. In MPNST with polycomb loss, you can see the loss of that methylation. And the polycomb loss is probably also the one that leads to poor survival. So you can kind of see that here from patients that have the polycomb loss mutation versus a different mutation, polycomb retained. So it could be a P53 or some other type of mutation. However, looking at histone H3 K27 trimethyl as maybe the marker for this is probably not the best thing to do, because K27 trimethyl is either over or underexpressed in a lot of different cancers, is kind of shown here, especially even cancers that are very difficult to distinguish versus MPNST, at least from a pathology kind of standpoint. And so starting this project, we had a couple questions. One, we kind of understand the first initial loss. You have this mutation to polycomb, you're going to lose K27 trimethyl, but how does that further kind of reprogram the epigenome? And that's something we wanted to answer with our quantitative epiproteomics profiling. And then, we are expecting big changes here in terms of the epigenome. So how does this affect transcription and essentially protein expression? And so we want to see what cellular pathways are affected by this polycomb loss. I've had two great postdocs that have been, John started the project, Joanna is currently working on the project. And so the first thing we kind of had to do was just convince ourselves that we can get high quality, proteomic data from formalin-fixed paraffin-embedded samples. And so oftentimes when there's a surgery, samples, these days you're getting a lot more samples being frozen, but historically samples were formalin-fixed paraffin-embedded in these little wax chunks. So we kind of had to work out a lot of the methodology to be able to get high quality histones and proteomes out of these samples. I'm going to kind of skip some of that, but it did take a fair amount of optimization, because this isn't kind of trivial to really retrieve protein samples from these FFPE blocks that have been sitting for 10 plus years. And so jumping into the data, if we took FFPE samples from patients that had just the neurofibroma mutation, ones that had polycomb loss, or one that had the polycomb retained, you can see there's something very different about the ones that have the polycomb loss. This is looking at the top 100 to 150 different histone modifications. So these kind of cluster together in this principal component analysis. You can see a little bit better in a volcano plot. So these are all the histone marks that are upregulated in the patients with polycomb loss. These are downregulated. The downregulated, they all make sense. They're all the K27 methylation-containing peptides, especially di and trimethyl. From the neurofibroma to the polycomb-retained, polycomb-loss samples, you can see that there's a loss of K27 di and tri. But there's other things that are either up or down regulated, and we kind of seen that. There's other histone marks, such as the H4 K20 methylation that's down regulated, and then you have all these methylations and acetylations that are upregulated as well. These circled right here, they're all histone H4. And so in blue is the polycomb retained. In red is polycomb loss. So when you lose polycomb, you have higher levels of these different histone H4 acetylation isoforms, as well as H3 and K36 methylation as well. This is kind of really interesting because originally we thought, well, this must mean that K36 methylation levels are increasing. That's only partially true. Actually, what we ended up finding from the data was that there's peptides that have K27 monodi and trimethyl along with K36, for example, dimethyl. In the polycomb retained cells, if we look at the total levels of K36 dimethyl, they're pretty evenly, or I shouldn't say evenly, but they're found often with K27 monodi and tri, as kind of shown here, unmodified monodi, tri, K27. But in the disease state, you lose the K27 methylation, and so basically you just have K36 alone with the unmodified K27. And so it's not so much de novo synthesis of K36 mono and dimethylation, but it's actually losing the K27 that kind of then we see the K36 increasing. And so what this data is telling us is that we're kind of going from a state where you have K27, the silencing mark, both by itself and maybe even with some active histone marks, which is in itself kind of really interesting that you would have the silencing and active marks together. But in the disease state, you lose a lot of the K27. It's a dominant global dominant negative loss across the genome, so you have mostly just open active histone marks found. We did see that K27 dimethyl and K27 trimethyl are both downregulated MPNST, and that's really interesting in itself as well because in other cancers I told you that K27 trimethyl is downregulated, but in some of these other cancers K27 dimethyl is not. And so these two are very specific markers for MPNST, and John, who is actually a resident pathologist / postdoc, has gone back to the clinics and been using these two marks to differentiate MPNSTs versus the K27 trimethyl alone. So we know what happens to the epigenome. You go from kind of a more silenced, closed state to more open, active state based on the histone marks. What about the proteins or the gene expression? And so if we do quantitative proteomics, top 6,000 proteins or so, another volcano plot, polycomb loss versus polycomb retained, you can see there's a lot of things that are upregulated in the polycomb loss. And when we do go-term analysis, they're all proteins that are involved in keeping an open, active chromatin, SWI/CIF remodelers, as well as proteins that are involved in transcription. And so this matches really well with the histone data. So we go from a closed chromatin to more open, active chromatin, and the proteome supports that. We have proteins that maintain open, active chromatin, maintain transcription. But as you see here, we have a lot of proteins that are downregulated as well. So what are these proteins? They're all proteins that are involved in the immune response. So MHC protein complex processing, interferon signaling, all things that essentially kind of potentially could lead to a patient being a little bit more immunocompromised. And so, if you just even look at the MHC class 1 and class 2 proteins from patients with polycomb loss versus retained, you can see a high level or higher level of these MHC protein groups in the polycomb retained versus the polycomb loss. You can see this just by bird's eye staining, histochemistry staining. So you can go polycomb retained for both of these HLA molecules. Higher staining goes to lower, higher staining goes to lower. And so the proteomics data kind of tells us that the histone marks there's more active open histone marks, and there's a subset of the proteome that's also upregulated as well, mostly involved with keeping an open active chromatin, but then the immune response antigen pathway, presentation pathways are downregulated in these patients. And so the question now arises, well, you lose a repressor such as polycomb, this silencing protein. I understand how you can maybe, lead to, activation of genes that were once, targets of polycomb, but how do you get more repression by losing a repressor? So that was kind of interesting. And so John kind of went back through the data, and what he found was that this protein, DMNT3, it's a DNA methyltransferase, was also upregulated in the patients that had the polycomb loss. And in fact, these patients have hypermethylation. When we did a global DNA bisulfite sequencing, these patients had higher levels of DNA methylation. We also see this by quantitative proteomics. So we've developed a methods to be able to do be able to analyze nucleic acid modifications. And so we see several modifications, methylations and hydroxymethyls, and even some DNA damage markers that are all upregulated in these patients as well. If you look at where in the genome this upregulation, hypermethylation is found, so you have the polycomb-repressive patients here, red is low, blue is high. So you go from low methylation to high methylation, you can see that you have higher methylation in the patients that have the polycomb loss, and go-term analysis is that finds that they're all on the promoters of things that we saw being downregulated in the proteomics experiments, the interferon pathway, all the immune response type of proteins. And so the picture now kind of is that, well, you have polycomb loss, it leads to open active chromatin and some gene expression, this is expected, but there's, immune response is downregulated, and the mechanism we think for that is that when you lose polycomb, you have DNA methylation, which is another silencing mark taking over and silencing the promoters of these genes. So what happens if we manipulate these pathways? What if we restore polycomb activity in these patient-derived cell lines that have the polycomb loss, or if we knock down other enzymes of these histone marks that are upregulated, or if we inhibit DNA methylation? And so I kind of told you before that maybe in the normal state you have K27, a silencing mark, along with K36, an active mark together. In the disease state, you lose the K27 methylation, so you have basically the K36 or some of these other histone active marks found alone. What happens if we restore the mutated subunit, SUS12 is what's mutated in this complex, and try to get the methylation back, or if we just remove the K36 and get to this state? So we can knock down NSD2, it's an enzyme that methylates K36, and what if we can get to this state? Is this good as well? And so we can, at least in patient-derived cell lines, we can add back wild-type SUS12. And when we do that, we can actually IP out the entire complex together, which is something we couldn't do with the mutated SUS12. So if we try to pull out the complex by pulling out the catalytic domain, we can't pull out the entire complex in the patient-derived cell lines that have the mutation. But when we restore SUS12 wild-type, we can pull out the whole complex. And when we restore the whole complex, the methylation levels come back. So the mono, di, trimethylation levels all come back. We can knock down NSD2, so we can get to here. So this kind of gets us to this point where we restore it. We can get to this point right here if we knock down NSD2. And when we knock down NSD2, we knock down the K36 methyltransferase. So we've kind of created this state and this state. So what happens if we look at the quantitative proteomic profiles of the SUS12 add-back or the NSD2 knockdown? It's basically the same genes. A lot of genes that we saw before being affected, such as the interferon genes and other transcriptional repressor. But basically, this is saying that these two different histone marks are found on the same genes, on the same genome, same parts of the genome. How susceptible are these cells to the knockdown or removal of the K36 methylation? Well, in patient-derived cell lines that have a different mutation, the polycomb-retain, these cells are not sensitive to losing NSD2. But in the patient-derived cell lines that lose NSD2, that lose polycomb, you can see how sensitive they are to losing NSD2. And we could do this trick of removing histone methylation across the genome by putting in this transgene. It's the same story. These cells that have a different mutation don't care if you do this. The cells that have the polycomb loss mutation, they do care that you lose the K36. So it's just a different way of doing. Instead of the knockdown, we remove the modification. So this could be a therapeutic vulnerability in the future. With DNA methylation inhibitor, well, you're going to increase or you're going to kill any cells that have DNA methylation and inhibition. So if you increase the concentration of DNA methylation inhibitors, you can see how much more sensitive or you can see how sensitive even the cells that have polycomb retained are. You can kill them, but look how much more sensitive the cells that have the polycomb loss are to the DNA methylation. And when you add a DNA methylation inhibitor, you get re-expression of all those interferon genes, which kind of confirms what we thought was the mechanism, where you lose polycomb, DNA methylation comes in and silences the immune response. You lose polycomb at these targets, you actually get upregulation of genes that were downregulated before. However, the problem by doing these manipulations is that it's fine in cells, but to restore protein activity in a patient is difficult, and there's no NSD2 inhibitor. And so these two are not really viable potential therapeutic options. DNA methylation inhibitors obviously are. But there's a lot of other histone marks that are differentially expressed, especially acetylation. And histone deacetylases and DNA methyltransferase inhibitors may share a mechanism of action. And so what we thought we might be able to do is kind of push the cells to a hyperacetylated state so that it could, potentially induce toxicity. And so we could do this by using these inhibitors. In polycomb-retained cells, when we add an HDAC inhibitor, we get increased H3 and H4 acetylation. This acetylation levels are already higher at a baseline level in patients that have the polycomb-retained. And when we add the HDAC inhibitor, the cells really go high in terms of their acetylation levels. So we do feel we're getting to this point. And in terms of their growth curves or kill curves, when we have an increasing concentration of an HDAC inhibitor, panobinostat, you can see that cells that have a different mutation, not the polycomb mutation, this is essentially what they can tolerate in terms of the increasing concentration of the HDAC inhibitor. These three in red all have the polycomb mutation. These are much more sensitive to increasing concentrations of an HDAC inhibitor. And then when we add the HDAC inhibitor, we can get re-expression of all of those HLA molecules that we had repressed by in the disease state. We can play that game where we add the wild type SUS12 back, and you can see that this shifts the ability of these cells to tolerate more of the HDAC inhibitor. But when we use a combination of increasing HDAC inhibitor and a single dose of DNA methylation inhibitor, you can see that the two together are more potent in killing the cells than essentially just the HDAC inhibitor alone. And so with this quantitative point of view, we've learned a lot about this system and what's going on in terms of epigenetic reprogramming and how this could lead to progression of cancer. We published an initial paper, several years ago, but a lot of the data I showed you was actually new in here as well. Really quickly, I wanted to share this new project. It's kind of in process, so definitely not a complete story. But we always kind of had a question of if you had a highly modified histone tail and you wanted to remove all the modifications, like how do you do this efficiently? You'd have to bring in all the demethylases and deacetylases that would come in and remove these modifications, and that seems like a lot of work. But we always thought, well, if you just want to remove all these modifications, what if you just clip this tail? And you can remove all the modifications, maybe the histone would be exchanged. And there could be a lot of consequences here. If you had modifications and readers and other proteins that were attracted to these tails, but then you had proteolysis event, you could kind of remove that, remove all these interactions. And in fact, histone proteolysis has been shown in many different contexts to be involved in a few different processes, especially cancer progression. One of the proteins or proteases that has been identified as a histone protease is cathepsin L. It's been found to cleave histone H3 during mouse embryonic stem cell differentiation, and so to cleave, and has been actually found to cleave histone at specific sites on the protein. There's also been cathepsin has also been found to cleave histone H3 during senescence as well, as a way to remove tails that have methylation to silence genes. And so there's a few papers here and there. Cathepsin L, though, is kind of interesting. The L stands for lysosomal, so it really shouldn't spend a lot of time in the nucleus. But there are papers that show it can cleave histone H3 and also cleave transcription factors, and this affects differentiation as well. And so you can see here, cathepsin knockout mouse, it affects keratinocyte and the mouse color and hair and things like that. And so we thought, well, are there any other cathepsin L substrates in the nucleus? I'm going to focus on histones, but we have a lot of data on other substrates as well. And so what we actually found when we looked back through some data that we had where we took intact histone, didn't chop it up with proteases, just threw it in the mass spec directly, was that we found a lot of cleavage sites for several histones, but especially this is actually histone H2A. So we saw a lot of cleavage fragments of histone H2A. And then we took just recombinant histone H2A, incubated it with cathepsin L, and saw that there were some sites that were highly preferred, which matched, some of the in vivo data that we saw, especially kind of like G34 and L23. And so we decided to kind of look at if there was any processes that, had a differentiation of proteolysis, these histone fragment events over a time course. And we went to mouse embryonic stem cells as our current model. These can be differentiated in many different lineages. It's already shown before that cathepsin-L is active in embryonic differentiation. So we want to kind of revisit this with a quantitative point of view. And so we can differentiate cells with retinoic acid to make embryonic bodies and eventually neuronal-like cells. And what we found when we did, some initial westerns is it's kind of hard to see, but you can see that, there's a lighter kind of fragment band of histone H2A that's lighting up here. With our quantitative proteomics and looking at the fragments, we can see that during a 4-day differentiation, we get higher levels of several histone cleavage sites or fragments, especially L23 during day 4. So in fact, there is cleavage and there are fragments that is occurring during stem cell differentiation. So is cathepsin, the protease that also cleaves histone H2A? We thought so because there's a little bit of similarity in some of the major sites that we saw cleaved on H3 and H2A. Cathepsin itself increases during differentiation, its expression, so it's actually the mature form is smaller, it goes through some processing, but you can see at day 4 it's pretty high. And so we knock down cathepsin in mouse embryonic stem cells, and when we knock it down, we can see several sites that go down, especially this L23 site decreases in abundance when we knock down cathepsin versus the control. Some other sites don't change, so there might be other proteases that affect them, or maybe they're just processing artifacts of what we're doing. But there's some sites that show differential cleavage when we knock down cathepsin. So why would you want to kind of, why would you need this protease to cleave? As I mentioned before, if you have modifications on a histone, it's a great way to remove the entire tail, remove all those modifications. When you look at H2A, H2A has several lysine residues that can get acetylated. They're kind of lower level, but they do get acetylated, K5, K9, and then the combination together. And over differentiation, you can see that the abundance from day 0 to day 2 goes up for all of these, and then it kind of dips at day 4. And so what happens when we knock down cathepsin here during this differentiation? Well, in the control, at day 4 it goes down. I just showed you that before. But when we knock down cathepsin, the levels don't go down anymore at day 4. And you can see for the K9 and then the combination mark, the levels drop. But then when we knock down cathepsin, the levels don't drop anymore. So this kind of told us that cathepsin is the protease for histone H2. Oh, I think I'm frozen. Oh, there we go. And so we wanted to see where K9 acetylation was in the genome, turning to ChIP-seq, both in the control and the knockdown of the cathepsin. And essentially here what you can see is that from day 2 to day 4 in the control, there's these peaks that have H2A acetyl that decrease a little bit, right? You can kind of see this almost a little bit on each one. It's kind of hard to see. But when you knock down cathepsin, you can see those peaks do not decrease, and in some cases they go up, which is what you would expect. And so essentially, when you knocked out cathepsin and compared day 2 to day 4 of the acetyl peaks, a lot of them that went down in the control do not go down anymore. This is just kind of showing you a visual of what's summarized here. And the expression levels from day 2, or the number of peaks from day 2 to day 4, you can see them going down here, but they increase here. Go-term analysis showed that they're all things that are involved in cellular differentiation, especially to the nervous system. So if, not a lot is known about acetylated H2A. Not very many papers are known, but our whole idea or hypothesis is that if you're cleaving H2A, it's probably because there's something recognizing those modifications on the tail. And so we thought to do an experiment to try to identify what might be recognizing acetylated H2A. So we just took synthetic peptides, either with or without the acetylation on H2A, and just did pull downs from nuclear extracts, did quantitative proteomics where we compared H2A acetyl, H2A K9, or K5 acetyl, K9 acetyl, or the combination together. In all of these three, we pulled out proteins, a lot of proteins that were similar, especially BRD7, BM1, SMARC, CB1, and for those that might recognize these, this is a PBAF complex, so this is a switch-SNF complex. There are several domains such as the PBRM or the or the BRD7 that have bromodomains. They might actually bind these acetylated sites, and this is just kind of validation. If we use an unacetylated H2A, it doesn't pull down these members as strong as if we have the acetylated H2A. Definitely pulls them down. And as I mentioned, several of these proteins are part of specifically the PBAF. There's different switch-SNF complexes, but this is all units that are specific to the PBAF complex, and PBAF is known to play a role in maintenance and self-renewal of pluripotent stem cells. So we can kind of see potentially that, if you have the H2A in embryonic stem cells, the switch-SNF could be binding that at specific genes that need to be turned on to maintain a pluripotent state. But once you differentiate, maybe you cleave the tail, PBAF comes up, the gene regulation or gene pattern, expression patterns are downregulated, allows the cells to differentiate into different lineages. So we wanted to look at the overlap between K9 acetylation and a couple of the members of the PBAF complex, ARID2 and PRBM. So again, ChIP-seq. As you can see here, these are the peaks that have the ARID2 or peaks that have ARID2 or the H2A-K9, PRBM1 by alone or PRBM1 with the H2A-K9. And as you can see here, the box that increases the most is at the promoter. And so you get a stronger or a larger number of peaks that have both ARID and the K9 or the PRBM and the K9 at the promoters. And you can kind of see that there. In terms of the expression, it matches really well. The expression of genes that have ARID is not as strong as the expression of genes that have ARID and H2A K9 acetyl. And the same thing, genes that have PRBM, the expression is not as strong as genes that have PRBM and the H2A K9. And so, kind of wanted to do some experiments to see what would happen if we kind of manipulate and can we create clipped H2A containing nucleosomes, and what does that do? And so we express full-length H2A or clipped H2A into cells, and then the flag IP. The flag IP showed that the clipped H2A is missing a lot of proteins that we saw before, especially the PBAF. And there's actually proteins that are upregulated in the clipped H2A. These two in particular we're following up because we think these are proteins that are involved in removing the clipped H2A. So these are exchange factors or nucleosome kind of assembly factors. So we think these might be the proteins that are actually recognizing a clipped H2A and removing them. And when we have these clipped H2A nucleosomes, we can see when we compare the full length to the clipped H2A, the full length can bind PRBM quite well. The clipped cannot, is what you would expect. And when you do ChIP-seq experiments, you can see here that in the control knockdown, these are the levels of the PRBM. When we knock down cathepsin, you can see these are the levels. They're actually a little stronger for these three, for the histone mark and these two clipped versions. And if you kind of just pull out a track, you can see here when we knock out cathepsin, the peak gets a little bit stronger from just the H2A. And here as well, you can see PRBM, you're looking at this one right here. So in the knockdown of cathepsin, we get stronger H2A, K9 acetyl peak, definitely a stronger PRBM peak, and a stronger ARID peak. And so we feel that the clipping of H2A is influencing the ability of PBAF to be found at specific genes in the genome that have the H2A. Lastly, last slide of data, essentially here, with these clipped or full-length H2A, we can see that the histone marks on them are quite different. The clipped H2A actually is found with more active genes, which is what you would expect, right? Because we know that it's getting clipped because it has probably more acetylated H2A, but we also see a greater amount of acetylated H3 as well, and also other marks which are more active marks, whereas the full length has enrichment of histone marks that are more linked to gene silencing, these K9 modifications. Lastly, the clipped H2A, if you're getting clipped, you could imagine it can't stay in the nucleosome forever. It's got to be a little bit less stable, and it definitely is. So if we do a pulse chase type of experiment, and we look at the incorporation of the full-length H2A or the clipped H2A, we can see that once we add cyclohexamide as well as we've used some proteasome inhibitors as well. In some experiments, I think these do not have it, but these are just inhibiting protein synthesis. You could see that the levels of the full-length H2A kind of stay for several hours, but the clipped H2A here, it's gone, or it's not as stable as a full-length H2A. So that's kind of a work in progress, but a different mechanism we think could be involved in regulation of gene expression during differentiation through the PBAF complex, recognizing some low-level histone H2A acetylation. And so with that, showed you a little bit of some of the technology advances as well as a couple different projects, one that's a little bit more mature where we've kind of followed the epigenetic kind of changes that occur because of these mutations in the soft tissue sarcoma, MPNST, and that gives us a lot of targets that we're now trying to get data in mouse models and hopefully keep marching towards a potential clinical therapeutic intervention. And this other project's a little bit more on the basic science side, but we think that proteolysis by cathepsin or other proteases could be a mechanism to regulate histone modifications as well, doing it in a much more permanent kind of way, and it may be important during stem cell differentiation. So with that, I'd just like to thank the group. This is the current group right here. A lot of great people that have contributed to these different projects, especially Joanna is working on the MPNST, and Francisca is working on one of the people working on the clipping project. Rick and Emily are people that have been working on the technology, or at least what I've showed you. We have a lot of other technology advances as well that we're working on and funding from different places, especially the NIH. I'd be happy to try to answer any questions if you have any. Thank you.
Looks like we have some questions. Please, in the hall. Okay.
Yeah, so Mark Nupper from NHLBI. I have a couple of technical questions. The work describing the quantification of the N-terminal post-translational modifications of histones is extremely interesting, but as you pointed out, it's also very challenging with the number of modifications, the density of modifications, and the number of possible combinations. I wonder what your thoughts are on the relative merits of top-down analysis versus bottom-up analysis. And secondly, what's the thinking with regard to bottom-up analysis as to what protease to use? I would think with all of the lysines, trypsin might be problematic.
Yeah, no, it's good questions. And so I think it really gets back to everything I kind of showed you was bottom-up mass spec, essentially, smaller peptides. But it just really depends on the questions you're asking. And so if you're asking about combinations of modifications, you want to look at a bigger stretch, you're going to have to do middle or top down. And so there's challenges there. Bottom up is much faster, much more quantitative, easier to do. Top down, it's basically the opposite of that, right? And so, yeah, we've done a lot in both worlds, but I think to answer biological questions right now, most people still prefer bottom-up because you can kind of get at what's happening. If you do see something that might be hinting at a combination, like, for example, we saw a peptide that had both K27 and K36 methylation changing. And so I know I didn't show you the data, but we wanted kind of to extend that and see, like, what other methylation. So we have done some top-down on like the MPNST project to see does it extend to other modification sites, and it actually doesn't. So it's a really kind of local binary kind of combination. So it really just depends on the question, but definitely if you're going to middle and top down, you better be prepared to spend a lot more effort on that. What was the second question? The protease. So I didn't go over the complete platform of what we do, but for bottom up, what we normally do is we chemically derivatize the histone so that we block lysines. So if it has a methylation or acetylation, it'll be naturally blocked. But if it's unmodified, it can get cut by trypsin. So we chemically modify it to block the unmodified lysines. And then we digest with trypsin to just hit the arginines. And the arginines, thank God, are like nicely spread out. So you can actually get, with this approach, 80 to 90% sequence coverage across all the histones.
Cool, thanks.
Yeah.
Okay, please.
Yeah, great talk. So I might have missed it, but the cathepsin is being targeted to all of the histones that have certain modifications, or do you have any idea what is targeting it to specific ones?
So a previous paper that I kind of mentioned does show that it can hit histone H3. But from our own data, it seems like H2A, we see more abundant fragments. So we think H2A is the main substrate, but we do see a little bit of H3 being cleaved even in our samples. And we even see a little bit of H2B being cleaved. So it may be that just that once it targets there, it prefers H2A, but it's, promiscuous and kind of hitting a little bit of everything.
But it's been targeted to certain parts of the genome, or?
Yeah, so I think so, because it looks like it's mostly affecting histone H2A patterns that have the acetylated H2A, and that's not everywhere in the genome. So how cathepsin is getting targeted to that, we have no idea. But it seems to be targeted to that H2A acetylated form, which is a very small, probably like less than 0.5% of the entire H2A has those marks. So there is something very specific about it getting targeted, but we have no clue what that is. We've tried to do some like cathepsin IPs and see what's binding to it, but nothing that makes a lot of sense, is coming down. Maybe we need to use cross-linkers or something like that so we don't lose whatever it's interacting with. But yeah, that is a good question. Like, I don't know how it's getting targeted to the genome.
I have also a question about cathepsin, but more general, because I didn't know that how unique it is that it is a protease going into the nucleus, and can it be a more general phenomenon about other proteases? You hinted in the conclusions that there may be other proteases working on histone.
Yeah, I mean, there's probably about like 5 or 6 papers published on histone cleavages or proteases. Cathepsin's one. There's at least 2 or 3, or 1 or 2 other proteases that other people in different organisms have seen get into the nucleus and cleave histones. So I don't think cathepsin is the only one. We've done some top-down kind of nuclear, looking at intact proteins from the nucleus, and we see a lot of fragments too, so of other proteins. And so I don't know if it's cathepsin or might be, I think there could be a lot of proteases getting into the nucleus and specifically into chromatin.
Right, yeah, very interesting. Thanks.
Okay, I think we'll conclude. Thank you very much.
Great. Thank you, everybody.
There's a reception just outside for those who are here. We can't keep the food until September, so please enjoy.